Hello, Assalamualaikum and welcome to our channel. Today's our topic is blood grouping or blood typing. What is blood grouping? First of all, see the simple definition of blood grouping. RBCs express various kinds of proteins and carbohydrates on their cell membrane as antigens. Means RBCs have various kinds of proteins and carbohydrates that they express on their cell membranes, and that carbohydrates and proteins are used to classify them in the groups. And there are basically two main grouping systems. One is ABO system and other is RH system. Now, before proceeding to the blood grouping, we have to see what are antigens. If you see the definition of antigen, you will see that antigens are molecules that are capable of stimulating the immune response. Means you have various molecules in your cells, but some of the molecules in your cells are capable of stimulating the immune responses in the other bodies. If that particular molecules enter into a different body that they don't belong to, then that body will stimulate their immune response against those molecules. So all those molecules that are capable of stimulating the immune responses in other bodies are called antigens. So what are antigens? Those particular molecules that are capable of stimulating the immune responses are called antigens. Means the antigens are identity cards of our body. If you remain in your body with that identity card, nobody will harm you. The cells will recognize you with that identity card that you belong to our own body, and no cells which are in our defense line will harm those cells that are with that particular identity. But if some other cells with some different identity cards, means if different antigens will enter our body, so the cells will recognize them as a foreign invader and will immediately attack them. As all cells, our blood cells also have that identity cards, means antigens. And with those antigens, our white blood cells or our defense cells recognize them as our own cells and don't attack them. And with those antigens that are actually the proteins or carbohydrate molecules that are expressed on the cell membranes of the RBCs, RBCs are grouped, or the blood grouping is made, or blood typing is made. There are two grouping systems. One is ABO system and other is RH system. In ABO system, that is derived from a parent carbohydrate molecule that is called H antigen. This is H antigen. If you have just H antigen on your cell membrane, your blood type is O. But if your H antigen has an attached enzyme that is called N-acetyl galactose transferase, that is attached via a molecule that is N-acetyl galactose amine, your blood group will become A. But if that H antigen is attached with the galactose transferase via galactose molecule, your blood group will be B. But if your H antigen is attaching both the molecules A and P, means N-acetyl transferase and galactose transferase, then the blood group will be AB. That means you will have a constant H antigen, means whatever your blood group is, you have H antigen in your body. Okay, now see the chart of antigens and antibodies of ABO system. If your blood is O, means no antigens are present in your body, nor A, not B, then your body will make both antibodies, antibody A and antibody B. Now come to the Rh blood grouping system, that is most complex grouping system and second most important after ABO system. It has highly polymorphic genes that encode them. But the most important gene is antigen D. If antigen D is present in the blood group, that is Rh positive, and if it is not present in the group, it is Rh negative. When people are given blood transfusions of the wrong blood group, the antibodies react with incorrectly transfused blood group, and as a result, the erythrocytes clump up and stick together, causing them to agglutinate. The coalescence of the small particles that are suspended in the ice solution, these larger masses are usually precipitated. Red blood cells agglutinate, clump up, clog. This is IgGM that is naturally present in our body and has 5 different sites which attaches the red blood cells, and red blood cells seem to agglutinate with each other. That's all for today. We will discuss Bombay type blood coming video. Till we meet again, Khuda Hafiz. If you have antigen A in your body, in your red blood cells, your body will not form the antibodies of antigen A. What are antibodies? Antibodies are a defensive molecules that will attack that particular antigen that is entering in the body. Means if antigen A is present in your body will not form the antibodies for that antigen because you have that antigen in your body cells, but your body will form other antibodies that is not present in your body. And antigen B is not present in your body, so your body will form antibody B. And if you have antigen B in your body, then your body will form antibody A. If you have A and B antigens in your body, and your body will not form any antigens against the red blood cells, means you have no antibodies in your body.